Good afternoon, everybody. My name is Michiel, and I'm here to present Plaza, and more specifically, the pan-genome analysis toolkit that has been implemented within the Plaza platform within the context of the EPAN project. So, of course, first of all, what is Plaza? Plaza is a platform for comparative evolutionary and functional plant genomics, acting both as an analysis platform and as a data warehouse. The first version was created in 2008, containing only 9 species, and the latest version was published in 2022 with 135 plant genomes. This latest version is implemented in 2 different instances, both focusing on separate clades, although there are, of course, shared genomes such as outgroup species and model organisms as well. And you can visit Plaza at the URL indicated there. So what can you do with Plaza? So first of all, we start from a gene-centric approach. We start from the protein coding genes, which are automatically clustered into gene families. And then through the use of data workflows, we can create then additional data types, which can be explored through visualizations and analysis tools, such as orthology-related information, phylogenetics, functional annotations through the appropriate ontologies, and collinearity and synteny. Additionally, because people often work not on a single gene, but rather on gene lists, such as differentially expressed genes from an RNA-seq experiment, we also offer the Plaza Workbench, where people can analyze entire gene sets at the same time, and then perform, for example, bulk orthology mapping or geo-enrichment analysis as well. Finally, although most people use Plaza through the website, there is also an API available, and for most of the data types, there are also file exports available as well. So, as indicated, when we started with Plaza in 2008, there were only 9 genomes available. In 2022, we had to make a selection of 135 genomes out of a total of more than 500 available genomes at the time. And now, only 4 years later, in 2026, there are already more than 1,000 plant genomes publicly available. This, of course, poses quite a challenge from a data integration perspective. This is, of course, further complicated because we are talking here only about publicly available genomes. Private enterprises will have sequenced their own genomes as well. And also, we are only talking here about the green lineage as well, which, of course, is a limiting factor. So, in order to deal with this, we have been working in the past couple of years on Fringe. So Fringe is our new technology, which allows people to create their own Plaza-like instances on their own hardware or in the cloud, if they so choose to, using their own genomes or a combination of their own genomes with publicly available genomes. This tool is not yet fully functional, but will be released later this year. So, circling back to Plaza, what we did was we first started looking at some recent publications and looked at, okay, what are those people researching, what are their goals, and so forth. Then based on that knowledge, we developed some new data types and associated visualization analysis tools, and then we tested it on 4 different pan-genomes that were integrated into Plaza. So these are the 4 pan-genomes. We start from Arabidopsis thaliana, pretty limited pan-genome from 2019. It's also one of the earliest one in plants. And then we added 3 major crop species as well. The Orchideum vulgare one is still being computed. It poses some computational challenges. But you also see that for Orchis sativa and Solanum lycopersicum, which are rice and tomato respectively, we have created these instances. Now, they can be accessed publicly through these QR codes, and these presentations will become available after the webinar in the training material, so you can look at it over there. So what did we do, of course? What features did we implement specific to pan genomes? Well, first of all, we implemented the well-known pen core plot and the core shell analysis plots, as indicated before by in the Pon-Barlex talk. So what you can see here, for example, are the genes that are either present in all genomes of the pen genome displayed in blue, which is the core section of the pen genome. And then you have the dispensable section, for example, where genes are only present in 2 or more species, but not all. What is important, though, is that all these charts and visualizations are fully interactive. So by clicking on them, you will be redirected to the associated gene lists. And then these gene lists can be either exported, loaded into the workbench, and so forth. To go a bit more in depth, we also implemented an upset plot. For the people who don't know what this is, this gives also a more in-depth view compared to the core shell plot from the previous page. So what you see here, for example, on the left-hand side, this 9,745 gene families, or the gene families present in all the different genomes of the pen genome. While on the right-hand side, in green, you can see that you have the gene families that are present in only selected species. In the middle, in gray and in red, you can then see the gene families that are only present into a subset of the combinations. Now, these tools are, of course, fully interactive. So as indicated in this animation, you can click on it. You see the associated gene families table, and then you can get the associated gene list as well. Additionally, as you can see, for these gene lists, the associated geo-enrichment is also computed on the fly, which can be further explored as well. Now, these are all pretty basic, but we wanted to go further, of course. So to do that, we created a new metric, which captures the conservation of synteny in the window around each gene, and this is what we call the synteny score. Now, briefly, what it does is it looks around a window around each query gene and compares this window to the window around each ortholog of the query gene. So, for example, as you can see here, we have 4 different genomes, and around each, we have a window of 6. This is our context window. Then we can compute a score based on the overlap, where each color indicates a different gene family, which is shared or absent between them. And then we can average over them to compute our final synteny score for that gene. This synteny score has been proven to be scalable, deterministic, and quite resistant to outliers as well. So what can we do with this? So the first thing we can do, of course, is just plot them for each chromosome. So in this case, we plotted for chromosome 1 of the Columbia ecotype of Arabidopsis thaliana. And here you can see that, on average, we have a very high score, almost 1, because a score of 0, a synteny score of 0 would indicate that there is literally no synteny conservation for that gene, which can be because the gene is, for example, a private gene, or because it's been translocated through an entire different region, or if the score is 1, then it means that it's fully conserved between all ecotypes. So what we can see here in this example is that the score is overall very high, almost 1 everywhere, except in the centromere, which is to be expected. And these synteny scores are also colored as well, where the blue indicates that it's present in all genomes, while orange indicates that it's only present in a single genome. So, which is then indicating that even if a score is low, but is blue, that means that it's still present in all other genomes as well, but that the window around it is not fully synthetically conserved. So if you now compare this between different cultivars of Arabidopsis thaliana, for example, then you can see already quite some interesting things popping up. For example, here in the bottom 2 ones, circled in red, we can see that there are some huge valleys, let's call them, present, indicating that these might be regions of interest because there has been genomic rearrangements there. Although, of course, yeah, the genes are still conserved. What we can also do then, for example, is create a histogram. So on the x-axis, you can see that these are the bins of the different synteny scores, while on the y-axis, you indicate the number of genes present within each bin. So what we can see then is that in this example, it's for tomato, that the overall pattern is pretty much conserved. The average synteny score is around 0.8 or 0.7, which is pretty high, but you can also see that there are almost no genes showing fully conserved synthetic regions. Additionally, you can also see that the purple line is a bit of an outlier, as it shows, especially to the left-hand side, a significant fraction of genes showing very low synteny scores. This plot as well is fully interactive, so what you can do is select a region. So this gives a list of bins, which can then be clicked on, where you can then provide the gene list. And then this gene list is once again fully exportable and so forth. Now, if you compare the synteny scores between different pangenomes, then you can see that there's quite some variation there as well. So for Herbopsis thaliana, which is the top left one, and Orchis sativa, which is the middle right one, you can see that for Herbopsis thaliana, the average synteny score lies around 1, and the centromeres are very clearly indicated. While for Orchis sativa one, yeah, there is quite a lot more variation there going on. This is especially also true once you look again at the histograms. For Arabidopsis thaliana, there's a very significant fraction that shows very, very high synthetic conservation. For Solanum lycopersicum, this is still pretty high, while then for Orchis sativa, you see quite a lot more variation there as well. It's important here to note that QC, of course, still plays a very important role, because if there is genome fractionation or bad annotations, this will, of course, have a major impact on the detection of these synteny scores, or the computation of these synteny scores. What we also did, of course, was computing or automatically detecting structural variations. This is an example of Orchis sativa for 6 cultivars of chromosome 8. And here you can see that, yeah, there is quite some synthetic regions, because these are not only currently the regions that we have indicated, but we also can show inversions and translocations. This can all be done on the fly. We can also rearrange species on the fly and so forth. And not displayed in this animation, but if you click on a region, then the associated gene list will also be made available. So to conclude, the Plaza platform has been extended to include a very diverse set of pangenome analysis tools. We have only showed here a limited amount of these tools due to time constraints. Furthermore, we also want to note that the Fringe tool will allow people to create their own Plaza-like instances. And more importantly, all these pangenome developments that we've developed will also be available within the Fringe framework as well. And if there are any questions, I will be happy to answer them during the Q&A. Thank you.